By this point, we've talked about the basics of the chemistry of life. We've gone over some basic definitions and some basic concepts in chemistry that help determine the properties of elements and molecules. We've then gone over what water is and why it's so important in life. And finally, we've gone over carbon and just why it's so special and making up the processes of life. But most of what we've talked about so far is pretty small stuff. And now we're going to start combining all of these things. And now we're going to be talking about what are known as biological macromolecules. Because most of the stuff that makes us up, it's not these smaller molecules like water or carbon dioxide, where it's just a couple of atoms put together. It's these large, complex molecules built around carbon and hydrogen. And for this video, we're going to be reviewing the four major classes of these and just how they work and why they're important. All of the molecules that we're going to be talking about in this section are based around what are called carbon skeletons. Now, remember back to our last video, carbon is really interesting because it has these 4 unpaired electrons that can then pair with other atoms. You can think of this as basically slots that are free and open for carbon to bond with. Because of that, carbon can actually form these really complicated backbones based on where multiple carbon atoms can link up into these long chains, and then other spots on them can be where something as simple as a hydrogen molecule to whole complex molecules in their own right can bind with. All of the molecules that we're going to be discussing have these carbon skeletons as their very literal backbones, either in the form of long chains or frequently the ring shape. Let's talk about the main classes of biological macromolecules. These differ in their overall structure, and from there, whether they are hydrophilic, dissolving easily in water, hydrophobic, not, or whether they can be both, depending on other parts of their structure. The other important thing that is something that carries over across these different types is this idea of monomers and polymers. Many of these molecules are made up of multiple smaller molecules. In these, the overall large structure of the molecule is based on the assembly of these smaller units, much like a string of beads can become a necklace. All of these molecules can, in some way or another, be found in monomer or polymer forms, and sometimes in what's called dimer forms. I'll point out different examples of these types of arrangements as we go through. First up, we have carbohydrates. This is a term that you've probably, if nothing else, encountered on the back of a food label. Carbohydrates are important because they are what we get our energy from. The simplest carbohydrate that our body uses is glucose. That is what is actually broken down to produce ATP, the molecule that drives energy in our cells. But while glucose is a monomer form of a carbohydrate, these get much more complex. You can have larger carbohydrates, frequently in the form of dimers, where it's 2 molecules of another type of a given type of sugar stuck together. Regular table sugar, sucrose, is an example of this. Or they can be found in much larger, long chains of carbohydrates. These are often structural in nature. For example, cellulose, which is the molecule that provides much of the structure for most plants, particularly trees and other woody plants, is a carbohydrate, as is chitin, the substance that forms mushrooms and some parts of some insects. Meanwhile, in the liver of many animals, another polymer carbohydrate is a source of glucose, glycogen. This stores glucose monomers in a form that can be easily accessed and broken down when necessary. Our next group of macromolecules are lipids. These are a really broadly defined group that include lots of relatively small molecules, by biological standards at least, that are dissolvable in nonpolar substances. This means that for the most part, these either don't dissolve in water and are repelled by water molecules, or they are, but maybe not as effectively as they would be in a nonpolar solution. The most familiar type of lipids, although far from the only, are the fats. And these are probably the lipids you're most familiar with. These are defined as having a long hydrocarbon tail. In general, fats, which with the more scientific name of fatty acid, are a long tail of carbons with bonds to hydrogens that is then attached to a group of molecules at the ends that give it the acid part. This acid part is known as a carboxylic acid group. This means that fats have one end that is hydrophilic, but then the rest of it is this hydrocarbon that is hydrophobic. These are the most familiar types to us because these and the details of their structure are probably actually terms you have run into before, even if you didn't know exactly what they mean. Fats are further characterized by the nature of that hydrocarbon tail. In its simplest form, the carboxylic acid group is attached to a string of carbons that, with the exception of the ends, are bonded to the next carbon in the chain, and then two hydrogens off each side. This is the simplest form of that hydrocarbon tail. And these are what's known as saturated fats. Saturated, in this case, refers to the fact that all of the possible spots on that hydrocarbon tail are taken up by bonds with simple hydrogen atoms. How is this different from unsaturated fats? Well, remember, carbon atoms have 4 places for other atoms to latch onto. In the simplest form of the hydrocarbon tail, each carbon just shares 1 electron with each other carbon in the chain. In unsaturated fats, though, 1 or more of those pairs of carbon actually shares 2 electrons and has a double bond. This means that some of the spots that could be taken up by hydrogen atoms aren't. These are further broken down into monounsaturated fats, which just have 1 double bond, or polyunsaturated fats, which have 2 or more double bonds along that chain. Saturated fats are what you probably think of as a lot of animal fats, although it is certainly not exclusive to them. Whereas our mono and polyunsaturated fats are more frequently found in plant sources. This is what gives an avocado its creaminess and deliciousness. As I mentioned, there are other categories of lipids, and these vary substantially, and we can't go into the same level of detail for all of them. But other prominent classes of lipids include steroids, such as cholesterol, waxes, which you can imagine very similar in some regards to fats, and phospholipids. This last category is important because phospholipids make up the membranes surrounding our cells, and we'll be talking quite a bit about them when we do our unit on cells in a few weeks. Next up, we have probably the most varied class of biological macromolecules, the proteins. You'll notice on the picture that I posted that while our lipids and our carbs are nice little molecule drawings with all the atoms labeled out, for proteins we got this big ball of yarn. This is because proteins are big. Proteins also are interesting because while we've been looking at the structure basically at one level so far of where all the atoms sit, because of their complexity, proteins actually exhibit structure on multiple levels. Because of this, proteins can fill many different functions in living things. Proteins are responsible for building many parts of your body, but they act as signaling proteins, and they act as enzymes that break down other proteins or build other proteins. Really, most of the time when you ask, well, what class of molecule is doing something in a living thing, most of the time the answer is going to be a protein. As I mentioned, proteins have multi-level structure. At the basic structure, we have what's called the primary structure. This is the sequence of amino acids the subunits of a protein or peptide that are held together in a certain sequence. For instance, here we see the primary structure of one type of protein. These are held together with what are called peptide bonds. So sometimes you might hear the word peptide thrown around, and sometimes they mean protein, sometimes they mean things made of amino acids that aren't quite as complex as protein, but that's what that word is referring to, things bonded together in this particular fashion. Now, think back to our discussion on water and how different ends of the molecule can have different charges. This is the case as well with the amino acids. Different parts of those amino acids have different charges, basically. Some might be slightly positive, some might be slightly negative. So if you can imagine having a string of magnetic beads, those may interact in very unpredictable fashions to form weird shapes that you don't expect. And this is, at its core, what's going on with proteins. This initial string of amino acids all bound together will then start to form what's called the secondary structure. This is where those ends of those different amino acids start to interact and twist this strand in interesting ways. The two main forms that you'll see are the alpha helix and the beta sheet. And again, these are held together with hydrogen bonds, much like water and some of the other substances we've talked about in this unit. That gets us to the initial shape of that strand, but even these shapes aren't the end of the structure of protein. Next, we have the tertiary structure, and this is where we get that lovely bowl of noodles look that so many proteins have when they're depicted. This is how, again, those charges and those bonds interact together to form complex structures. Then is determined not only by these sorts of interactions, but by the specific amino acids, then interactions with the outside molecules, specifically water. So tertiary structure is not only based on how the polypeptide chain that is forming the primary and secondary structures interacts with itself, it's then being shaped by how it interacts with the environment outside of the protein itself. And these are often mind-bogglingly complex, requiring massive amounts of computational power just to figure out and render. And this, for some proteins, like myoglobin, the simple one that helps bond oxygen in your muscle cells. Some of them end at this tertiary structure, but many proteins are not even just one strand of these amino acids. For these, we have the quaternary structure, how these different strands called subunits interact and sit together to actually perform the function of the protein. These can get quite complex. The one shown here is involved in some aspects of DNA replication and is formed of 3 protein subunits. Hemoglobin, the protein in your blood that binds oxygen, is composed of 4 subunits, but many other types can be composed of far, far more. It's these interactions and large-scale structures that make proteins so complex and allow them to perform a mind-boggling array of biological functions, from basic structure in your cells to signaling to more exotic things like the toxins in different venomous and poisonous animals, for instance. Our final class of biological macromolecules are a bit more complicated in some ways than what we've talked about before. So far, we've talked about things where the monomer units are fairly simple. They're an amino acid, or they are a fatty acid, or they are a sugar. But for our last class, the nucleic acids, our monomer unit is a bit more complicated, as each nucleotide, as it is called, is made of 3 components in and of itself: a 5-carbon sugar, a phosphate group, and a nitrogenous base, which is just a fancy word for a organic compound that contains some nitrogen. We will get more into the details of what those bases are and what they do in Unit 2, where we talk more specifically about DNA. Because, as you might expect from that little spoiler, the 2 main nucleic acids that we will be talking about are DNA and RNA, and these are primarily used in
encoding the information needed to build basically all of the others. DNA is the double-stranded version, and RNA is the single-stranded. I'm not going to cover these in quite as much detail here because we have a whole unit going into DNA coming up, but they are a distinct type of macromolecule in and of themselves. This wraps up our videos for this week, talking about the chemistry of life. Hopefully, you now have a better understanding of just how the atoms and elements in the world around us work together to form the biological molecules that we rely on and are made up of. As we go through the material in the coming weeks, think back to these concepts to connect how the chemical basis of life plays into the other aspects of biology that we'll be discussing. See you next week.